Hey everyone, and welcome. Today we're diving deep into one of the most fascinating chapters in all of biology, the molecular basis of inheritance. We're talking about the actual story of life's code, the instruction manual that's written inside every single living cell. All right, let's get right into it. You know, for centuries, people knew that traits were passed down, that kids look like their parents. But the how was this huge, profound mystery. I mean, how can a bunch of molecules store, copy, and then pass on the entire blueprint for a living thing? That's the big puzzle we're going to solve together today. So, here's our game plan. First, we'll kick things off with a big quest to find the genetic material. Then, we'll get into what DNA actually looks like and how it's packed. After that, we'll see how it's copied, how it's read to make proteins, and finally, how that code is controlled and used in the real world. Let's begin. All right, so our first stop is basically a scientific whodunit. Back in the early 20th century, scientists knew that the instructions for life were tucked away on chromosomes. The problem was they didn't know which molecule was the real star of the show. Was it the complex proteins or was it this other molecule, DNA? The search was on. So, the first big break in the case came way back in 1928 from a scientist named Frederick Griffith. He was working with two different strains of bacteria. One was a deadly S strain that caused pneumonia, and the other was a totally harmless R strain. And the experiment he did revealed something, well, pretty mind-blowing. Okay, so the first three steps, totally predictable. You inject the deadly strain, the mice die. You inject the harmless strain, the mice live. You kill the deadly strain with heat and inject it, the mice live. No surprises there. But, and this is where it gets crazy, when he mixed the harmless live R strain with the harmless dead S strain and injected that, the mice died. Somehow, the living harmless bacteria had been transformed into the deadly kind. Some kind of transforming principle had been passed from the dead cells to the living ones. But what was it? Well, for the next decade, a whole team led by Oswald Avery worked to pinpoint exactly what that principle was. They basically set out to identify the mystery molecule. They took the heat-killed S strain bacteria and painstakingly purified all the different biochemicals from it, the proteins, the RNA, and the DNA, to find the culprit. So how did they crack it? It was this brilliant process of elimination. They took the extract from the deadly cells and treated it with different enzymes. First, they added an enzyme that destroys only protein. Did transformation stop? Nope, it still happened. Then they added an enzyme that destroys RNA. Still happened. But then, and this was the absolute game changer, they added DNA, an enzyme that chews up only DNA. And just like that, the transformation stopped dead in its tracks. And boom, the verdict was in. It had to be DNA. Still, some in the science world were skeptical. They needed one last final nail-in-the-coffin piece of evidence. And that came in 1952 from Alfred Hershey and Martha Chase. They were working with viruses that infect bacteria, and their idea was pure genius. They used radioactive tags to label either the protein coat of the virus or its DNA core. Since DNA has phosphorus but not sulfur, and protein has sulfur but not phosphorus, they could track each one separately. So here's what they did. They let these tagged viruses infect the bacteria. Then they put them in a blender, a regular blender, to shake the virus particles off the outside of the cells. Finally, they spun everything in a centrifuge to separate the heavy bacteria from the lighter virus coats. The big question was super simple. Which radioactive tag actually made it inside the bacteria? And the results? They were just crystal clear. The only time radioactivity showed up inside the bacteria was when the DNA had been labeled. The radioactive protein coats just stayed on the outside. This was it, the final, undeniable proof. DNA is the genetic material. Case closed. Okay, so we've found our molecule, DNA. But, solving one mystery just opens up a bunch of new ones. Like, what does this amazing molecule actually look like? And how in the world do you pack meters of this stuff inside a nucleus that's too small to even see? Let's go investigate the architecture of life's code. So let's start with the basics. The fundamental building block of DNA is something called a nucleotide. You can think of it like a little three-part Lego piece. It's got a sugar, a phosphate group, and then one of four nitrogenous bases. These are the famous letters of the genetic code, A, T, C, and G. These little nucleotides link up together in a long chain, creating a sugar-phosphate backbone, which gives the whole thing direction. And that directionality is absolutely critical. And let me tell you, these chains get insanely long. A simple virus might have tens of thousands of base pairs. A bacterium like E. coli has about 4.6 million, but a single human cell, it has about 3.3 billion base pairs. If you stretched it all out, you'd have almost 2 meters of DNA packed inside a single tiny cell. That's just wild, and it means there has to be a super clever way to get it all to fit. Then in 1953, Watson and Crick came along and blew everyone's minds with a double helix model. It's made of 2 long chains that run in opposite directions. We call that anti-parallel. The sugar phosphate backbones are on the outside and the bases point inwards, pairing up like the rungs on a twisted ladder. And here's the absolutely crucial part. The pairing is specific. A always pairs with T, and G always pairs with C. That's the rule. Okay, so here's a classic question. Why does the DNA ladder have perfectly even steps? Why is it so uniform? Well, the secret is in the size of the bases. A and G are bigger molecules called purines, while C and T are smaller molecules called pyrimidines. Since a big purine always pairs with a small pyrimidine, the total width of each rung stays the same. It's a perfect fit every single time. So, how do you solve that 2-meter-long string in a tiny cell problem? You use some very clever packaging. The DNA, which has a negative charge, is wrapped around a core of positively charged proteins called histones. This structure, DNA, wrapped around 8 histones, is called a nucleosome. It's kind of like beads on a string, and it's the very first level of getting all that DNA packed in. Now, these nucleosomes don't just float around. They get coiled up even more tightly into something called chromatin, and this chromatin comes in 2 main flavors. You've got euchromatin, which is loosely packed and ready for action. Its genes can be easily read. And then you've got heterochromatin, which is super tightly packed and silent. Its genes are turned off. So you see, the packaging isn't just about cramming it all in. It's a super smart way the cell controls which instructions get read. Okay, so a genetic material has to do 2 things, right? Store information and make a perfect copy of itself. The amazing thing is the very structure of that DNA double helix gave scientists a huge clue about how it might do just that. And what's so cool is that Watson and Crick pretty much called it in their original 1953 paper with this legendary understatement. They knew that if A always pairs with T and G always pairs with C, then the 2 strands are complementary to each other. So if you just pull them apart, each side can serve as a perfect template to rebuild the other. And that idea leads directly to what we call semi-conservative replication. The original parent DNA molecule unwinds, and each of its 2 strands is used as a template to build a new complementary strand. What you end up with are 2 identical DNA molecules, but each one is a hybrid. It has one of the old parent strands and one brand new daughter strand. That's why we call it semi-conservative. It's half conserved, half new. Now, how do you prove something like that? With a really, really clever experiment from Messelson and Stahl. They grew bacteria in a medium with heavy nitrogen, so all their DNA was heavy and dense. Then they moved the bacteria to a medium with normal, light nitrogen and let them divide once. When they checked the DNA, it was all an intermediate hybrid density, exactly what you'd expect. After a second division, they found both hybrid DNA and purely light DNA. And that right there was the proof. Replication is definitely semi-conservative. Okay, so we have a stable code and we've got a way to copy it perfectly. But so what? How does the cell actually use these instructions to build things and run the show? This whole process is called gene expression, and it's basically the central dogma of biology. The first step in reading the blueprint is called transcription. This is where you make a temporary messenger copy of a gene, called messenger RNA, or mRNA. A special enzyme called RNA polymerase latches onto the DNA at a promoter site. It literally moves along the DNA, unwinds a little piece of it, and builds a matching RNA copy. Except it uses a base called uracil, or U, instead of T. When it hits a terminator sequence, it lets go, and you have your finished message. This mRNA now has the genetic code, and it's read in 3-letter words we call codons. Now, this code has some really cool features. It's degenerate, which means that for some amino acids, there's more than one codon that codes for it. But it's also unambiguous, meaning one codon will only ever code for one specific amino acid. And maybe the most amazing thing is that this code is pretty much universal, from bacteria to humans. It also has a specific start signal, AUG, and 3 stop signals. All right, step 2 is translation. This is where the message actually gets turned into a product, a protein. This all happens at a molecular machine called a ribosome. The mRNA blueprint is fed through the ribosome like a tape. Then another type of RNA, called transfer RNA or tRNA, acts like a delivery truck. Each tRNA reads a specific codon on the mRNA and brings in the one correct amino acid. The ribosome then stitches these amino acids together into a long chain, which folds up into a working protein. Now, a cell has thousands of genes, right? But it would be chaos if they were all turned on all the time. Cells need to be very smart about controlling which genes are on and which are off. And on our end, scientists have figured out some incredible ways to read this code, one letter at a time. The classic textbook example of this is the lac operon in E. coli bacteria. It's a group of genes that make enzymes to digest lactose, the sugar in milk. Think of it like this. By default, there's a blocker protein sitting on the on switch, so the genes are off. But if lactose shows up, it grabs onto that blocker and pulls it off the DNA. Now the switch is flipped on, and the cell can start making the enzymes it needs to eat the lactose. It's a super efficient system of supply and demand. Okay, now imagine going from regulating just a few genes to reading all of them. For a human. That was the Human Genome Project, this absolutely huge global effort to sequence our entire genetic blueprint. It was a massive success, giving us the sequence of all 3.3 billion base pairs. This project completely changed the game for medicine, for understanding human history, and for biology as a whole. And you know, understanding our entire genome isn't just for science textbooks. It's given us some incredibly powerful tools that we use in the real world every single day. Let's talk about one of the most famous ones, DNA fingerprinting. It's wild to think about, but 99.9% of your DNA is identical to mine and to everyone else's. But that tiny 0.1% that's different is what makes you unique. DNA fingerprinting hones in on specific parts of our DNA where short sequences are repeated over and over. The number of times these sequences repeat varies a lot between people, and that pattern creates a genetic profile that's unique to you, your DNA fingerprint. And the uses for this are just everywhere. It's a cornerstone of forensic science, helping to solve crimes. It's the definitive way to prove paternity. Scientists use it to track genetic diversity in animal populations to help with conservation. It can even be used to help diagnose certain genetic disorders. It's a technology that has truly changed our world. All right, so before we finish up, let's do a super quick revision. This is one of those must-know comparisons for any exam, so let's lock it in. Okay, let's nail down the key differences. DNA has the sugar deoxyribose. RNA has ribose. DNA's bases are A, T, C, and G. RNA swaps out the T for a U, so it's A, U, C, and G. For structure, DNA is that famous, stable double helix, perfect for storing information long-term. RNA is usually a single-stranded, more versatile molecule that does the active work, like carrying messages and helping build proteins. And there you have it. We've gone on this whole journey, right? From those early detective stories, figuring out what the genetic material even was, to understanding its beautiful double helix structure, and then seeing the elegant machinery of how it's copied, read, and translated into the stuff of life. So as you move forward, just remember, this stuff isn't just a random collection of facts and experiments. It's the fundamental language of biology. It's the blueprint, the shared code that connects every single living thing on this planet. Understanding this blueprint is the key to understanding life itself.